Faults in DNA molecules can be harmful, so all organisms have DNA repair mechanisms that can correct most of them. Mutations, that is, changes in DNA sequence, can appear in DNA either as mistakes in DNA replication, which results in a mismatched nucleotide pair, or by the chemical or physical action of a mutagen. Other agents can cause breaks in DNA molecules that may in turn lead to mutations. Most cells possess 4 different types of DNA repair systems. Direct repair and excision repair systems fix DNA molecules carrying nucleotides damaged by mutagens. Mismatch repair corrects mismatched but otherwise normal nucleotides that result from errors in replication. Non-homologous end joining, NHEJ, is used to mend double-strand breaks in DNA. There is also a 5th repair system called homology-directed repair, but it will not be covered here. Direct repair is the simplest repair process and acts directly on damaged nucleotides, converting them back to their original structure. Only a few types of mutagen damage can be repaired directly. In this example, guanine acquires an alkyl group, which disrupts its normal pairing with cytosine. In a future round of DNA replication, the alkylated guanine would pair with thymine, thereby propagating a DNA mutation. To fix this chemical damage, an enzyme transfers the alkyl group to itself, restoring the base to normal. Many organisms have enzymes that perform this function, such as the ADA enzyme in E. coli and MGMT in humans. In contrast to this simple single-step repair, most types of DNA damage require several steps to fix. They can be repaired only by removing the damaged nucleotides and then filling in the gap. This process is called excision repair. Base excision repair is a type of excision repair mechanism that is used to repair relatively minor damage. The damaged base is excised from a nucleotide by a specific DNA glycosylase, which first flips the damaged base out of the helix and then cuts the beta-N glycosidic bond between the base and the sugar, leaving a baseless site, called an AP site. An AP endonuclease cuts the phosphodiester bond on the 5' side of the AP site. And then the other 3' side is cut, either by the endonuclease itself or by a phosphodiesterase. The resulting gap is filled in by a DNA polymerase and sealed by a DNA ligase. The direct, base excision, and nucleotide excision repair mechanisms all act by searching for and correcting abnormal chemical structures in DNA. However, they cannot correct mismatches resulting from errors in DNA replication, because the mismatched nucleotides are not abnormal in any way. A special type of repair, called mismatch repair, is used instead. Rather than detect an abnormal nucleotide, the mismatch repair system detects the absence of proper base pairing between the parent and daughter strands. Before the mismatch repair system can excise part of the daughter strand and fill in the gap, it must first distinguish between the parent and daughter strands. In E. coli, the 2 strands are distinguished by their differing levels of methylation. The newly synthesized daughter strand is not methylated, but the older parent strand is methylated at GATC, CCAGG, and CCTGG sequences. In the mismatch repair system of E. coli, a protein called MUT-S recognizes the mismatched nucleotides, while another protein called MUT-H distinguishes the 2 strands by binding to nearby unmethylated 5' GATC 3' sequences. That is, by binding to the daughter strand. MUT-H cuts the unmethylated DNA at the methylation site. Starting at this cut site, a DNA helicase detaches a segment of the single strand. The detached single-stranded region is degraded by an exonuclease that follows the helicase and continues beyond the mismatch site. The gap is then filled in by DNA polymerase I and DNA ligase. Note that eukaryotes do not use DNA methylation to distinguish between newly synthesized and parental DNA. Instead, they use a different mechanism that is not yet well understood. A double-strand break in DNA is potentially devastating to a cell. Such breaks can be generated by exposure to ionizing radiation and some mutagens, and occasionally during DNA replication. They are repaired either by a process called non-homologous end joining or by homologous recombination, not illustrated here. In non-homologous end joining, a pair of proteins called Ku bind to broken DNA ends. The individual Ku proteins also have an affinity for one another, which brings them and the two broken ends of the DNA molecule into proximity. The DNA fragments are joined back together by a DNA ligase. Experimental studies indicate that restoring normal DNA structure is difficult to achieve. Often, the nucleotides flanking the double-stranded break are lost, resulting in a loss of normal DNA sequence. Alternatively, if two chromosomes happen to be broken, a misrepair resulting in hybrid structures occurs relatively frequently. Hence, this type of repair is often prone to errors, in contrast to direct repair, base excision repair, or mismatch repair, which restore normal DNA sequence with far greater fidelity.